Hello, everyone, and welcome to our cancer vaccines talk. I'm going to use this talk as a bit of a precursor to for our guest lecturer, which will be tomorrow, who is working at Dendreon. So we're going to talk a little bit about Dendreon's cancer vaccines. We'll talk a little bit about cancer vaccines in general and how they differ from traditional vaccines. We'll have another talk I'll record on kind of normal vaccines and immunodeficiencies coming up next. So this one will be a good one. Hopefully, it'll give us a little bit of background. I don't know exactly what our guest is going to talk about, but he might touch on some of these topics, so this might help for the presentation tomorrow. We've come to this slide in the past. We've talked about this before, but in terms of how your immune system recognizes a cancer cell as self versus non-self. And there are a couple ways in which this is accomplished, one of which is by presenting altered self peptides. So that's one. Expressing proteins at the wrong time. That's two. Or overexpressing genes. So these are kind of three traditional ways of seeing these types of things, and it brings up kind of our introduction to the major problem with developing cancer vaccines. Is one of the first things you have to do is recognize specific epitopes or specific markers that cancer cells will present that can be then identified by the immune system or removed. We're also going to talk a little bit about what cancer cells do to avoid the immune system, because if your immune system should already be seeing things like this, and they're not, and so cancer cells have done things to evade the immune system. So you kind of it's playing a game in a sense that cancer cells are using tricks, molecular tricks, to avoid detection by cytotoxic T cells. And so now how are you going to activate the immune system to instead recognize them? So here we are. So this chart shows a little bit some of the types of antigens that are expressed in cancer cells, and these are looking at embryonic proteins. So that's one of the things that was on the previous slide. And if you look at these cases, we're looking at AFP, which is alpha fetoprotein, and carcinoembryonic antigen, CEA. That's a mouthful. These are two kind of embryonic antigens. Again, these are things that are expressed at the wrong time. They're normal proteins, but they get expressed in tumor cells. And here you see in the first part of it, when you look at non-cancer diseases, they have no levels of these fetal proteins. But in carcinomas here, you do. You get 69% of patients that have this particular liver cancer. So this becomes a very solid marker for liver cancer here. Now, not all of them are as straightforward. If you look at CEA, in this particular case, now you see the different types of cancer here. So from lung cancer, pancreatic cancer, breast cancer, the percentage of patients. And what you see is quite a big scale. It's certainly increased over non-cancerous diseases. So it is a marker for cancer, but it's something you need to think about. When you're developing a therapy and you're developing something that's going to treat a lot of patients, you have to look at, well, if we're trying to treat colon cancer, yes, this is a good marker, but still only 35% of the patients are expressing it. So even though it is correlative, it's not as high as something like 70% that for this AFP. So finding a specific marker that is present in a high percentage of cancer cells is something that's very, very challenging, and it's one of the big challenges with developing any type of treatment for a cancer cell that's based upon recognition of a tumor-specific antigen. Now, if we look at, as I mentioned already, one of the issues is cancer cells do things to avoid the immune system. So the expression of altered antigens or high amounts of MHCs here activate cytotoxic T cells. When you have tumor antigens here, these altered antigens, that also, both of these ones, these guys are getting killed. So these are the normal mechanisms. Well, so if you're developing an immune therapy, you have to recognize that you can't just depend upon the cell expressing the particular antigen you want on MHC receptors, because one of the mutations that takes place as a tumor cell is mutating is it shuts down and decreases the expression of MHC receptors. So here what you see is the ability of this cell to escape detection by cytotoxic T cells. And these guys are the problem. So these are the cells we need to worry about, because our natural immune system is not getting to these guys. So when we talk about cancer vaccines, the whole goal is using T cells and trying to reactivate or engage the immune system to recognize and destroy these particular immune cells. Okay, here we are, cancer cells. So kind of repeating what I just said, the whole idea of a cancer vaccine is to activate the immune system, eliminate tumor cells. And these vary significantly, although we haven't talked about it quite yet, but everybody's aware of vaccines for infectious diseases, especially, everybody's had vaccines, whether it's for a flu vaccine, smallpox, chickenpox, whatever it is, they're protective. And the whole idea is really they're B cell, they're based on creating memory B cells. And so for these things, you're given exposure to an antigen, the disease, the virus, in a weakened state or a component. We'll talk about some of the different ways in which this works. But the idea is you're exposed to it, you activate B cells, you produce memory cells with high affinity for that, so that when you come into contact, you have antibodies already primed, ready to go, and you can fight off that disease. Cancer vaccines are the opposite. They are given after the diagnosis. So they are a treatment, not a preventative measure. And what they're really trying to do is, instead of creating memory B cells, you're activating T cells to eliminate and kill cancer cells. So it's the fundamental difference between the two ideas of cancer fighting. With that, I've already touched on some of these things. There are big challenges with developing these cancer vaccines. One, as we just saw, identifying antigens that are specific to that particular cancer cell. We've seen with these two kind of embryonic antigens, those are prevalent in many cancer cells, but they're not 100%. So you're going to only be treating a certain percentage of the population. It can be difficult finding antigens that are unique. It could be it could be overexpressing a normal protein, and that antigen might be found on other normal cells as well. And so then you get non-specific effects that can be very damaging to the organism, but humans, a person with the disease. You have the tumor the immune response is weakened in the cells that have this cancer. So the cancer cells is evading. We've mentioned some of this already. The immune these things have to be overcome as well. And then you can deal with things with autoimmunity when you're injecting different types of antigens, as I mentioned already. If they're not unique to the cancer cell, they could essentially create an autoimmune response where they're attacking cells that are expressing your antigen in a normal manner. So these things can be obviously really challenging. Here are some examples of tumor-specific antigens, and sometimes mutations can be the best targets if you can identify them, because they are expressed in tumor cells alone. And if you looked at things like tumor suppressor genes, anti-apoptotic genes, oncogenes, things that are mutations that are found in tumors, but not found in normal cells. So if you can figure out mechanisms to attack those specific antigens, then you have a viable mechanism to attack these antigens. This can also be challenging as well because mutations, not every person is the same. In terms of cancer, and we talked about this before, cancer is a multi-step process in which you are developing mutations in a series of genes, and you need multiple genes to develop cancer, and that's why every cancer is slightly different. So you won't have companies or researchers spending a lot of money for, let's say, a specific P53 mutation where there can be a lot of P53 mutations that are found in cancer, and so what happens if it creates a P53 mutation that is a different antigen than the one you want to target? So here are some tumor-specific antigens, and I should mention, I'll mention this again. This is actually, these slides I got a long time ago, actually, from another visitor from Dendreon who was working on their vaccine research, so it'd be neat to see some of the updates on it. But she gave me these slides. She was actually the head of their vaccine development department, and so it's a nice background, and they're that I've been using ever since. So it'll be interesting tomorrow to see if we can get any updates. Okay, so here were some of the tumor-associated antigens that have been identified. The oncofetal antigens we've talked about already. You can have tissue-specific antigens. Here, the one we're going to be talking about, this HER2/neu, is something that's expressed in breast cancer cells, and that's going to be the basis of the Dendreon's cancer vaccine. And then a lot of you have heard of the papillomavirus in cervical cancers, and there are vaccines, not to be confused with the types of vaccines we're talking about now. The vaccines against the papillomavirus to prevent cervical cancer are not cancer vaccines. They are normal vaccines. They're trying to prevent the infection of this virus. This virus happens to insert in the human genome at a common kind of hotspot that makes people susceptible to cervical cancer. It's a nuanced argument. We can talk about this more in class, but it's important to recognize that's a different type of cancer virus. So HER2/neu is something we're going to talk a bit more about, and that's what Dendreon is based or was basing one of its strategies on the breast cancer vaccine. One that could be in here too is prostate, which is the prostate-specific antigen that they worked on, and we'll see in this particular talk. Okay. There are different mechanisms for making cancer vaccines, and they really span. There are a lot of different techniques. I'm not going to talk about all of them now. We're going to focus on a different type. We'll kind of go over them briefly in a minute, but they're all based upon altering tumor cells to reactivate the immune response for them. So I'm going to show a slide in a second. We'll come back and look at this, where you see there are things like DNA, viral vectors, bacterial vectors, gene-modified tumor vaccines, whole cell tumor vaccines, heat shock proteins. I'll show basically what these vaccines are based upon, reactivating the immune response and figuring out a way to get the immune system, specifically cytotoxic T cells, to recognize these specific antigens. And we'll go into some more details in just a minute. What we're going to talk about with Dendreon's vaccine is this, what they say, the ex vivo administration of antigen to autologous cells. What that means is you get your own dendritic cells. You take them out of bone marrow, and then the antigen is delivered as a peptide to those cells in culture. So you grow those cells in culture and then have them express that peptide. Now that peptide has to be specific to a tumor cell peptide. So now when you put those tumor cells back in the patient, those dendritic cells, not the tumor cells, sorry, the dendritic cells back in the patient, those will then activate T cells to recognize a particular tumor cell. So that's the vaccine we're going to be talking about. So before we get into that in more specifics, here are some of the things. This is kind of the graphical representation of that slide I was talking about. Different ways you can take and alter, try to alter the patient's tumor cells so that they are better recognized by your immune system. So here's one where you're genetically modified. So you have a genetic modification. You take out tumor cells. So in here, each of these, you're taking out a tumor biopsy here. And in that case, you're extracting heat shock proteins. And so heat shock proteins are generally, remember, they're chaperones, and they're generally bound to unfolded proteins. And so the idea is in tumor cells, you have a lot of unfolded proteins that are bound up in, because of the mutations that are taking place, that are bound up and being held in these chaperones. And as a result, when you inject them back into the patient, then they can be kind of chewed up, presented on antigen presenting cells, and be markers to tag or to help your immune system recognize the kind of mutations and misfolded proteins that are found in tumor cells. Here you're taking tumor cells out, you're adding a particular gene, this GM-CSF, which is a cytokine we just talked about. That GM-CSF will be expressed by these tumor cells, which that will help activate immune cells around them. Here is actually the CEA antigen, so that embryonic antigen we talked about before, carried on a particular virus that you can inject into a tumor. So if you can inject into the patient, into the tumor, so that it now expresses the CEA antigen that you are guaranteed this tumor will have it, and it will help activate the immune response to it. Then there are other things where you can again look at dendritic cells that will recognize kind of hallmarks of cancer cells. In this case, they are transfecting dendritic cells with telomerase. That telomerase then is primed, presenting those peptides on the surface of the antigen presenting cell that will then get recognized by T cells. Cancer cells are generally immortal, so you need telomerases. That's another protein that should not normally be expressed, and so that helps them be recognized by the immune system. So you see, from this, again, this is from the visiting scientists that we had a number of years ago. These were companies that were working on them. Some of those, I'm sure, are gone these days, but you get an idea of the different characteristics. So to reiterate how these cancer vaccines work, what's happening is that you need to activate these cytotoxic T cells, so these MHC class I restricted cells. That's what you're trying to activate, and it's still, there are different types of these cancer vaccines that are being done, but there are different ways you can do it. And so we're going to look at, go into more detail at the Dendrion's vaccine, but there are many ways to do it. And so here's kind of some of the different mechanisms that's beyond. You have, here are, your cytotoxic T cells here, but you also can have strategies in which you're using antibodies to recognize. In this case, here's a breast cancer cell. Here's that HER2 new gene, which is overexpressed. So if you have antibodies that recognize it, and then using Fc receptors on macrophages or neutrophils can target that, your immune system can target that for destruction. You can have cytokine therapies that we were just talking about that can help assist in this as well. So there are different ways in which we do it. So let's go into some more specifics about different types of cancer vaccines. So here's one type of cancer vaccine. One is actually transfecting. Transfecting with the B7 gene. So in this case, you take a tumor cell, you transfect it. Again, you could do something like a biopsy we were talking about, or like the CEA antigen. It expresses that B7 gene. So think about that. It's an interesting idea. The B7, now you create this, essentially, these tumor cells kind of become antigen presenting cells, and they can essentially target themselves for you activate these cytotoxic T cells and activate and target themselves for destruction. So it's an interesting idea there. Another type of genetic modification we already mentioned in that kind of brief slide was the GM-CSF. So GM-CSF, you transfect a tumor cell with that, it produces that soluble cytokine GM-CSF, which will promote the activation of and stimulation of dendritic cells in the region around the tumor to promote destruction of that tumor. So those are, again, a little more elaboration on some of the ideas we already mentioned briefly in that slide. Let's look at Dendrion's idea. So Dendrion, as I've mentioned, one of the things the advantage they've done is worked on a particular, here's the name of it, Provenge, which is FDA approved now. PAP, which is prostatic acid phosphatase, it's found in 95%. So you can see it's a great candidate to develop this type of strategy or of a cancer vaccine because it's in 95% of all carcinomas or prostate cancers. They were also working on a HER2/neu, which is that breast cancer receptor, which is overexpressed. That one's only in about 30%, but that's still a very high number, and that's what makes them very good candidates. So they overcome that kind of first challenge. That first challenge, identifying your antigen, the antigen that is overexpressed. Now the question is, now what they did was create a delivery cassette. So the delivery cassette is what's interesting. And so in Provenge, it's PAP, and then they made a breast cancer one, which is HER2/neu. And so they made a fusion protein where you have your antigen specific to your cancer here hooked to a GM-CSF. So this essentially create a fusion protein, the GM-CSF, which is that cytokine here, which is the binding site. Remember, cytokines will bind to receptors, so you have a cytokine receptor, and then you have a peptide here. So what's the idea? Well, the idea, remember, this is these are autologous cell essentially transplantations, where you are going to get precursor antigen presenting cells from the patient. So you're getting that from the bone marrow. Then you add your antigen cassette, which has that GM-CSF right there, which binds to that receptor, and then through receptor-mediated endocytosis goes into the cell. And so now you bring that antigen in, it's now processed and presented on a precursor APC, or loaded, and then presented on a mature APC. So in this case, you have a dendritic cell, a mature antigen presenting cell that is from the patient, grown in what's called ex vivo. So you mature it outside of the patient in culture, and now these things can be given back to the patient, essentially given as a stem cell transplant or bone marrow transplant. And now they're preloaded with a prostate-specific antigen that should prime your immune system to attack the prostate cancer cells. So this was some of the actual clinical data that was presented a few years back. And what they did, and so there were some, as I mentioned, this is now approved now, but it gives you an idea of kind of the evidence of what you see here. That you can see a large solid tumor right here. Then post Provenge, that tumor has decreased dramatically. You can look at things with Gleason scores. Gleason scores just kind of tell you the aggressiveness of the particular cancer. And so in any particular therapy, I actually know a bit about this because my dad has prostate cancer. And he's actually in a clinical trial right now. And part of it of screening is that you have to find patients that have kind of similar types of cancer. So to quantify in a particular analysis or in a particular assay, you have to look at cancers that have, at least to the best of your ability, characterized and similar. Because if they're cancer cells, it'd be easy to bias your studies if you get patients, or you're treating patients with less aggressive cancers. And so what what every patient does is has to get it screened, and they have to have scores, Gleason scores, which identify them and make them applicable for this trial. As a side note, basically what happens in any clinical trial, it starts out with patients that are very sick toward the end of their life, where all other therapies have kind of run their course, and then they get to try some of these trials. So you'll see in the analysis when we look at when we look at what happens survival time, you're talking about increasing the lifespan, of 7% of the patients by 8 months. But so you're looking at, in this particular case, when you treat people, these are people that are very, at very advanced stages of the disease, and that with the Provenge, that they have a significant survival rate in over 6 months, 12 months, and 18 months. But again, this is the initial evidence, which then can go on to then allow them to try this treatment on people with earlier stages, and that's really the goal. If it's effective at the late end of it, then now try it on the early end, and maybe that has a much more beneficial approach. But it's never, trials are really set up to, you don't start with those patients, because if there are approved treatments out there, they will exhaust those resources first. So here you can look at, again, this kind of reiterates the same data of looking at what happens to disease-related pain in this treatment, and you can see that people that are on the Provenge treatment actually are experiencing much less pain, and actually they're living longer too, because at this point all of the placebo group has passed away. Okay, so I already hit that one as well, and so that kind of gives you the basic idea of these dendritic cancer vaccines. And so we might hear a little bit about this, or hear some of the work that they're doing now. This drug is approved, but some of the stuff they're working on now might be or slightly different level. You can look at some of the reviews, and these were the two from a few years ago that they were looking at the prostate cancer and the breast cancer vaccine. So we may be able to find out a little bit about those particular vaccines. Okay, that is it. We'll have another vaccine talk for next week as well, where we'll compare some of the more traditional vaccines and the way they work compared to this for next week.